Hello everyone, how are you all doing today? We will be starting this session in 10 more minutes at 5 p.m. Indian Standard Time. So thank you for joining, and the chat box is open for any questions. So I welcome you all to this session. Please post any questions that you may have, and we will be talking soon and sharing with you all about NGS in a moment. We will be starting here in 3 more minutes and we'll wait for participants to join in today. Thank you. So just as we are starting the program and I'm receiving questions in the chat box from some of you, I want to address that we will be addressing some of the research questions as well today in this program and talk about the different aspects of RNA-seq analysis. And if you have any questions related to both the training as well as the research program, please continue to post them in the chat. Besides myself, we also have our team present here, so they will be able to answer any kind of questions you have related to either training in bioinformatics as well as bioinformatics research or bioinformatics projects, which we are going to address at length today. So thank you again, and we are going to be starting the session in 1 more minute. And I'm also putting down my email ID in the chat for anybody who may have these specific questions, especially related to research, and you can reach out to me there. Thank you. Welcome to this free webinar on NGS data analysis. Today we will discuss about the upcoming training program and the various aspects of RNA-seq data analysis and how this field of bioinformatics continues to evolve. This special program has been designed for students, faculty, and researchers. In this program, we will focus on one type of data, which is transcriptomics data. This program will cover analysis of gene expression data with practical sessions and online tutorials. Anyone with a basic knowledge of cell and molecular biology will be able to conceptually understand as well as practically apply standard data analysis tools to process, analyze, and interpret complex transcriptomics data. Together, we will study analytical approaches for RNA-seq data. Using bioinformatics methods and databases, we will be able to use RNA-seq data to study gene and isoform expressions in various organisms across many conditions. So the omics logic programs are a result of close collaboration between Pi and Biotech, an innovative big data and analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the omics logic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. In the last Omics Logic Transcriptomics program, which we had in 2020, many participants joining us were able to successfully complete the program. And the feedback shared by these participants are encouraging for our team as well as those who continue to take these courses. The participant feedback also demonstrates that the learning is effective and even transformational. The training is offered using and leveraging the regular online webinars like we're having today, practical exercises and online courses. These resources provide a broad and detailed overview of processing, statistical analysis, machine learning tools, and single-cell data analysis techniques. The courses cover methods, expand on various terminologies, provide practical examples, and offer quizzes for self-evaluation, while we use the server platform for running pipelines and the Codeomics Logic platform for learning more about R and Python and its utility in understanding bioinformatics data. My name is Viksha Biswas, and I'm the Omics Logic Community Manager, responsible for daily interaction with thousands of users who are part of our community. Before I introduce everyone to the team behind Omics Logic and today's session speakers, we would like to know more about you. So right now on your screen, you will see a poll. So here are a few questions, what we want to learn from you about your understanding of NGS. So the first question is, what are your interests in learning NGS data analysis? And you can choose from the 4 different options that you see on your screen. The second question is, what research area, I'm sorry, it's written where, what research area you want to apply NGS transcriptomics data analysis to? So these are a few areas. If you see that there is an answer and you would want to apply to any other area which I have not mentioned beyond these 4, please put them down in the chat box so that we are aware that the different research areas you are interested in. So I see that a lot of you are interested in precision oncology, very interesting, as well as infectious diseases. So how much time will you be able to dedicate for the NGS data analysis training per week? Okay, so I think we are having 6 to 8 hours is the highest number. And what NGS data analysis research you are familiar with? So there's single-cell transcriptomics, non-coding RNA, mRNA, and I'm a beginner and would like to learn more. Okay, so I see that today we have a lot of you who are just starting out with learning about NGS data analysis, which is interesting, and we will be covering some of these important aspects as well. So I'll just leave on this poll for 1 more minute, and then we'll move ahead with the presentation. So if you have not yet answered, please go ahead with the poll, and I'll be ending the poll in a minute. Okay, thank you, everybody. I'm honored to have been working with a team that collectively have amazing experience in this area, including the other co-founders of the company. Dr. Alfred Tauber comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation, and has a long-standing recognition for his generous support of many international projects and research activities. One of these projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do here at Pine Biotech. And here you can meet the other members of our team, as well as Dr. Leonid Brodsky. In the upcoming 1-month period, you will have the opportunity to interact and collaborate with an international team of experts specialized in the areas of bioinformatics and data-driven research. Let me introduce you to the program mentors. We have Dr. Harpreet Kaur, who is our Omics Logic trainer and expert bioinformatician, and she will mentor sessions and guide participants throughout the programs. Her area of specialization is in cancer genomics. We will be hearing more from Dr. Kaur today. Dr. Mohit Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is our CEO and co-founder. His experience in research and academia has led to many research labs and university collaborations and has been recognized for the simplified approach to omics analysis for education. Now, I would like to invite Dr. Mazumdar to talk more about RNA-seq analysis and NGS data. Thank you, Dr. Mazumdar. Over to you.
Yeah, thank you, Bipsha. Let me share my screen. Hello, everyone, and thank you for joining this specialized session on bioinformatics, omics, logic technology. My name is Mohit Mazumdar, and in my current role, I'm working and collaborating with universities in Asia and Africa to introduce innovative curriculums on data-driven biological research at different levels of education. So during this short period, I've been honored to be able to collaborate and work with thousands of students and researchers around the globe. So this free webinar is conducted for participants and especially students and researchers and faculty in India and neighboring countries. We have invited all who have shown interest in learning about NGS transcriptomics data analysis and research using our research program and platforms. In the context of the upcoming program, the online training is a unique opportunity to not only learn about NGS transcriptomics, but to master data science skill sets and apply to an original research project within 2 months of training. So let's learn about this exciting field of research. So Bipsha, I'm facing an issue with slide transitions because I think the co-hosting thing. So can you check that? So as I was telling you that let's start with learning about this technology. And to do that, let's answer a few questions. So molecular biologists study a broad spectrum of topics related to regulations of cell biology, from DNA and RNA to protein expression and then protein-protein interactions. So next generation sequencing is a key technology which is enabling us at an unprecedented level of precision and efficacy in understanding genomic and transcriptomic regulations. So a next generation sequencer creates a digital file, right? And that digital file represents a sequenced DNA or RNA. So this file contains information where nucleotides are represented by letters corresponding to their bases, right? So a dominant theme in molecular biology is how variation in nucleic acid, which is the genotype, affects the biology of the organism, which is the phenotype. So a second major theme in the study of gene expression and this resulting chain is observed transcriptomics features of a studying sample. So what is a transcriptomic feature of a studying sample? So in next generation sequencing data analysis, where we call it like RNA-seq a lot of time, the purpose of this analysis is to map the short reads from raw sequencing files to the reference genome, and then study the abundance of RNA mapped to a particular region. So this abundance can represent the level of gene or the isoform expression, and then it can be compared between samples or group of samples. So that is an overview about transcriptomics and RNA sequencing. So I'm still facing issue with the transition. So in today's workshop, we will start with big biological data and the high throughput sequencing processes, which are focused on RNA-seq NGS data. So to know more about this today, In this session, we will go through transcriptomics resources. Then we will be talking about the mentor support, and we will be demonstrate to you the explanation of methods using multi-omics t-bioinfo platform. So one of the major objective of this entire methodology, starting from the courses, training, support, and then learning the techniques. The major objective of this research training is to enable the participants to prepare for real world application of RNA-seq and to be able to apply training to a research project. So the how the overall project works, it can be summarized in a few steps. So let me tell you about a few steps that we can take to work on a project. So how does it actually works? So it can start from framing a problem, which could be like a title of your project, and then identifying a data set. So that data set actually represents the research that you're interested in. It could be agriculture, like many of you have shown interest that was there in the poll, that in agriculture, applying it in agriculture and biomedical research or any other kind of research. So from identifying a data set and the references to preparing the data, next step is to prepare the data, process the data for analysis, and then perform the analysis. So once your data is prepared, the next step would be to perform your analysis and go in depth of that, what you are finding. So whether your process that you're following is statistically correct and logical, and then you're trying to interpret the results. So in this today's webinar, and as we are speaking about this, we will be having Dr. Harpreet Kaur in a short time to talk more about applying NGS transcriptomics research in translational, how it is playing a key role in translational research, which is a boom in the biomedical industry. So now let me show you the design and the curriculum of the first 4 weeks of the training. So the program will kick off on 2nd March 2021, and the session on that day will feature the orientation session with our Omics Logic trainer, Dr. Harpreet Kaur, myself as the project mentor, and along with us, Ms. Beepsha Biswas and the team of interns and research fellows. So during the first week, we will learn about processing of gene expression. Then we will learn about alternative splicing, and then we will learn about assays for mRNA detection and quantification. So after completing the introduction to next generation sequencing to study the RNA expression, next we will learn about quantitative and qualitative analysis of RNA in a sample, and then data preparation using next generation sequencing and preparation of table of expression from raw FASTQ files. So all of our omics logic training kind of includes resources. So these resources are assigned to each sessions to help student make the most out of this training. So in this first session, we will be introducing you to a lot of resources. So this Omics Logic program leverages our top rated online tutorials that are present in this educational platform. So that includes, so the program participants will have the access exclusive resources that are assigned for each session that are associated with the training. So along with this, there is a smart tracking of activity, and there are curated data sets, biological data sets that are designed for a great learning experience. So after this, we will be talking about the analytical platform. So this program and throughout this course, you will be given directions to access the t-bioinfo platform. And I don't know if you guys are able to see the animation. So we have to fix this picture. Can you just sign out and sign in with your own login? Beepsha, can you hear me? Okay. Sorry for this. Let's just try to figure this out because uh-huh, I'm still having issue. So just for a second, let me log in again. So the slide transitions can be smooth because each time I'm doing the transition, it's getting stuck. So there are no animations that could be played with this. So just let me sign in and join again. So Dr. Harpreet, you're there?
Yes, I'm.
Okay, Dr. Harpreet. So I'm making you the host and I'm going to join again. All right.
Okay, doctor. So, Bipsa, I have made you co-host.
Yeah, thank you, Dr.
Mm,
Harpreet.
Okay.
Okay, so can you see my screen now?
Yeah, we
Yes.
can.
Okay, perfect. So as I was telling you that throughout this course, we will be sending you the directives to access the TBA Info platform. So the TBA Info platform have the processing area and analysis area for RNA-seq data. So the platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipeline, and different stages of analysis are performed in different sections of this multi-omics platform. It's a quite comprehensive platform which is used for various sort of trainings. We are conducting oncology specialization trainings. We are conducting infectious disease specialization trainings. So these are some of the panels have been um, collaboratively worked out with universities like UCSF, who with whom we have developed this virology section. And so, as you see, there are several sections here. So, in this program, our focus will be on the transcriptomics section. So, in this transcriptomics section, we will learn about first we will learn about the data processing, and then we will be using the section under data mining, and we will there we will be learning about different algorithms, which machine learning algorithms and classification algorithms that are going to help us understand the meaning out of this big omics data. And again, like when we are integrating multiple data sets, like you talk about the non-coding RNAs and mRNA, and then other omics data, then we have a dedicated section for multi-omics integration as well, which is quite popular and gives a lot of insights that we were not been able to gain before. So and then we will be using, so the other platform that you saw, the TBA Info platform, that does not require any coding, require you to do any coding. So you can learn from a visual environment and learn about the logic of the analysis. So the focus here is to learn about the logic, that why these steps are used and what are the standard output from an popular algorithms that are used for RNA-seq experiments. And we will be learning more about it when Dr. Harpreet takes you there. So we'll be doing that in some time. But to tell you more about that, how we are utilizing this in a synchronous way so that you gain a deeper understanding of how this sort of analysis is done and applied in current research. So you start with the understanding of the theory, and then we go into the practical aspects of doing the experiment, and then after doing the experiment, we would want to do a visualization and we would want to do some downstream analysis. So we have exercises designed to explain the codes for these types of visualization in both R and Python that will be used in this program. So along with that, we will be sending you the video tutorials, and these video tutorials explains different aspects of, whether we are going to want to install Python, whether we want to, do it on the platform, whether how do we want to, learn about the different sections of the platform. So all those are already available and video recorded tutorials with us, and we will be sharing those with our participants when we start the program. So after that, the first introductory week, where we will be sharing you about the platform, getting you more familiarized with the resources, and you are comfortable about, exploring and going to the platform and having your own profile. Then the next week, the next class on March 5th, we will start with the analysis. And to talk about the analysis, we will be doing, after gaining a basic understanding, we will start with a data set. So there is a significant interest in sequencing, especially in the context of oncology. And that is due to its potential to advance diagnostics and personalized treatment. So that added with that, there is an improvement on understanding of the treatment responses and the cause and mechanism of tumor development. So all of these things are contributing in precision oncology. So in this session, using a breast cancer data set, we will be reviewing methods, how data is generated using next generation sequencing, and use a sample project, patient derived xenograft transcriptomics biomarkers for breast cancer, to practice generating a table of expression from raw FASTQ files. So from a publication, that is the data which is being published and submitted in a NCBI bio project, we will be using that data set, and we will be giving you the publication, and we will be having this session to describe that, and we will be start working on the data and understanding of how from raw data you can generate a table of expression. So in addition to the online courses, obviously all the participants will get access to the hands-on exercises and data sets for practice. These can be completed without any coding. So this will be by using the TBioInfo platform that will give you an interactive learning experience. So talking about the transcriptomic sequencing, so this is a transcriptomics pipeline that has a number of algorithms that you can see. So transcriptomic sequencing is a major advance in the study of gene expression, in the field of study of gene expression, because it allows a snapshot of whole transcriptome rather than a predetermined set of genes. So before that, it was microarray. We have panels, then we have set of genes, which were biomarkers. Now we can look at the entire transcriptome at a given point of time. To study this data, right? It's a lot of data. To study this data, we will learn about processing pipelines. So this is a processing pipeline. A processing pipeline that turns raw reads, means the reads that are coming from a sequencing machine, into structured data which is ready for analysis. So this platform is color coded. Color coded means that section wise you can differentiate that which section is for which kind of analysis. So this helps to establish a logical connection between theoretical aspects of data analysis methods and the practical steps that are needed to achieve a biologically interpretable result. So the pipeline outputs that we get, so once you are done with your once you run your analysis, and once you get from a table, from raw sequence to a table of expression, so the pipeline outputs are offered as tables graphics and interactive visualization, helping users to understand the way data can be used to explore or understand a biological phenomena. So we will also learn to preview and visualize the resulting data using principal component analysis. So this is a very popular algorithm, which is a machine learning algorithm, which is used to visualize uh, high dimensional data, multi-dimensional data. So to perform the example analysis, this will be also provided, that is also provided in the course. Student will need to have the access of the T-Info platform to do the assignment, and then you will get all these sort of hands-on practical aspect of doing the analysis. So in this entire training, we will also be using Microsoft Excel or any other software, equivalent software for plotting. So this would also require us to go to some of the NCBI, for example, this is a JBrowse, where you can visualize the alignment in much more detail. So after doing all of this in the first week, the second week, we will be learning about data preparation using NGS and getting the table of expression from raw FASTQ files. So in the next session, we will review important topics and keywords that are associated with RNA-seq transcriptomics analysis. So in this session, we will learn about working with table of expression to get a high-level view of RNA-seq data, and the participants then will get their first hands-on assignment in this session on the patient-derived xenograph dataset. And we will also be sharing additional resources, reading material for all the algorithms that you saw there in Transcriptomics 1. So this will be also a review session and an interactive session to know if you have any problems or if you have any confusion, if you're getting somewhere stuck. So things like that. And after this, in this week, we will start exploring the basic coding. So for those who are interested in coding, could join this, and we will be able to do this coding in our code platform. So in this, we will be doing from loading data to some of the downstream analysis. So basically, it would be a very basic overview of how to export files and how to load that data using the code platform. So this week, we will be learning how to visualize, summarize, and analyze data from processing pipeline. So in transcriptomics, like starting from March 12th, on March 12th, we will be moving from table of expression and the basic analysis to more statistical analysis. So this Transcriptomics 2 would be more about understanding at a statistical level that how your data is. I mean, what your data says and what are the noise, how we can remove the noise and find the meaning out of those, uh, mm, out of the experiment that we have got. So one of the limitations in this sort of high speed data generation is the amount of noise and amount of, non-essential data that we get. So one of the um, key skills that comes from this sort of experience is to be able to learn about your data and visualize it when you have generated it, so that your downstream analysis makes sense. So some of these techniques are used to study the variation. So we will be using gene expression matrix, then that can be used to study the variation. So some of these techniques are really easy. It's not that tough. Some of these techniques are easy to use and can be done in Excel. And we'll be going through that in our sessions, and which will be recorded and sent out via email. So we will be using either Excel or some table manipulation software, and some of the other approaches, which are more streamlined approaches, those will be used using the T-Info cloud. So in Transcriptomics II, during this week, we will be learning about more complex algorithms, and we'll be using some of the statistical algorithms like t-test. And then the online session that will supplement this course Transcriptomics 2 module, it describes a number of statistical techniques typically used for simpler and then more complex approaches to find differentially expressed genes. So once the data is standardized and prepared, we will learn about working the identified genes to arrive at a biological interpretation of results. So ultimately, it's not just about the data, but also how to prepare it. We want to gain a biological our objective here is to gain a understanding or a biological understanding of the project. So connecting the data elements with their biological significance will be a major focus of this program. So in the third week, when we have this understanding of how statistics work, what is there to look into the data, we will be focusing on differential gene expression. So one of the major objective of doing transcriptomics research is sometimes and most of the time is to learn about the differential gene expression. And we'll continue to look at typical challenges that can be resolved using other methods. So we will use a t-test, then we will use DSeq2 to run a differential gene expression pipeline. And then we will use, in the next session, we will learn about the first machine learning method, which is the factor regression analysis. So it's a regression based algorithm. So in this session, after learning this, as a result, you will be able to detect obvious differences between preset groups, as well as expand the idea of more subtle differences presented by factors that might interact with each other. So when I'm talking about multidimensional data, then you will see that data has multiple features. So if you get a data for example, for a liver cancer sample, then you might find that uh, there are different factors that influences liver cancer. So it's not so simple that there is a direct connection, but there are multiple factors that influences one connection. And to understand about that, the statistical analysis that will be used is factor regression analysis. So, as I was telling you, that we will be working on data sets throughout this program. So, we'll discuss the property of such data sets, where thousands of observations about each sample contributes to noise and misleading data. Some aspects of log normal distribution and adjusted p-value, they are accounted for. False discovery rate will be explained in detail. So for our next session on Friday, March 19th, that will be dedicated to advanced analysis methods of transcriptomics data that allows us to find meaningful pattern in big data sets, especially complex patterns that are very typical for RNA-seq experiments. For example, PCA can help us understand variability between samples by analyzing all of our data features. So we can use such dimensionality reduction techniques to get a better insights into our data set and to be able to visualize it. So by comparing multiple methods and by plotting them, we will also learn about we can study genes. So as a result, we can study genes and how they describe how they're described in samples. So samples are maybe the patient samples in our data set, and how this correlates with phenotypic information we have for biological or even clinical interpretation. So we will learn about impact of feature selection on interpretation of conditions sampled on various data sets. So after exploring different methods for identifying groups of samples without prior knowledge, so when we don't have a prior knowledge, we call it unsupervised machine learning. And when we have a prior knowledge, we call it supervised machine learning. So one of the example of unsupervised machine learning is clustering. So in week fourth, we will examine methods for developing classifiers for known samples, which is predictive analysis or classification. So in the next week, after learning about unsupervised machine learning, we will be learning about approaches such as machine learning and data mining and predictive analysis using algorithms to learn about how they connect and how they give us what important information that we are asking from the data set. So to do that, we will cover decision trees and random forests to work with various gene sets for classification. For example, a defined pattern of expression can be linked to, regulatory network affected by a condition. So this could be one of the things that you get out of this sort of research. So, for example, we will learn to utilize gene set expression and gene set enrichment analysis to find pathways or gene set enriched, which are like either upregulated or downregulated and are genes of our interest. So during this entire exercise of learning about machine learning algorithms, we will be learning about dimensions, classes, and features, which are helping us understand how these machine learning algorithms can be applied to offer interpretable results. So application wise, after you saw this is like a journey, you start from basic understanding, basic data analysis, then you move towards the second area to more advanced statistical analysis, and then you move to a further advanced analysis of using machine learning. And now that how this entire research field can be applied to various areas of research. So sorry. So these methods are not always used in smaller research projects. However, they have much more important application in both pharma and biotech R&D, as well as in clinical applications. So mastering these methods will help you understand, for example, in clinical application or in biomedical research, you can think about biomarker discovery and patient stratification, both of which uses predictive modeling and feature selection. So here today, we have a very specialized person, Dr. Harpreet Kaur, with us. She has done her entire PhD and have published amazing papers on some of the aspects of NGS data and machine learning. So now I'd like to pass it on to Dr. Harpreet to guide us and talk more about bioinformatics projects and application of NGS data, and that what we will be doing in the second phase of this training in the week 5 onwards. So over to you, Dr. Harpreet.
Thank you, Dr. Mazumdar.
And yeah, please feel free to ask questions regarding whatever I have shared. So I'll be happy to answer them in the chat, and we will have a Q&A session towards the end as well. Thank you.
So, hello everyone. My name is Harpreet Kaur, and I have a PhD in bioinformatics. In my current role, I'm Omics Logic Bioinformatics trainer with Pine Biotech. I am assisting participants to learn about data analysis, machine learning, and visualization in the application to the last data set used in the biomedical research. Besides, I'm involved in designing different research projects using publicly available data. So the upcoming Omics Logic program will be focused on transcriptomics. We will discuss the various aspects of RNA-seq data analysis and how this field can be applied to the various aspects and continuously growing in the healthcare. So during this program, you will learn about how you can design your own independent research project based on publicly available NGS transcriptomics data and employing various bioinformatics techniques. So today we will perform various NGS data analysis using the data sets based on patient-derived xenograph project. So now let's understand what is a PDX project or PDX model. So a PDX models are generated when a fresh tissue obtained directly from the patient and is implanted subcutaneously and orthotopically into the immune deficient mice. So as these models maintain the principal histological, clinical and molecular characteristics of the original patient tumor samples while remaining biologically stable. Since these PDX models are more closely resemble and recapitulate the tumor growth in human than standard in vitro cell line. So they remain key experimental platform for preclinical drug development. So today we will try to analyze two breast cancer subtypes using PDX model. The breast cancer types include ER positive and triple negative. So this ER positive breast cancer have a special type of receptor, that is estrogen receptors, while the triple negative breast cancer lacks all the three common receptor on its membrane. So this is just a one difference that makes triple negative is a more aggressive form of breast cancer, while ER remains a more treatable form. So now let's go over to the server and let's run an example pipeline where we will learn how we can generate a gene expression, well-structured gene expression data table from raw sequencing read data. So let's move over T-band for server. So Bipsa, if you share this, okay, thank you. So on this interface, you will find various sections for different types of analysis. Like already Dr. Mohit has shown you, we have NGS data analysis, then mass spec analysis, then structural biology, then data integration and modeling. So under the NGS data section, here we have a subsection that is dedicated to the transcriptomics. So once you are on this server, so here you will find a different analysis pipeline. So today I'm going to demonstrate a simple example pipeline that is from the PDX project. So on here under the RNA-seq and chip parallel analysis of NGS and microarray data, here if we click on the drop down, you will get this demo RNA-seq PDX project. So I'm clicking on this as this is a demonstration pipeline. So we have already uploaded example data set, that is raw sequencing data. So by clicking on the start, we will able to upload this data. So here we have already provided the reference genome file, and as well as we have created the SVL links for the FASTQ files, and we have submitted that files here. And once we click on OK, we able to upload these files. So then the next option that is getting highlighted, that is under the pre-processing. So the next option which getting highlighted here is Trimomatic. So what it does, it will remove adapters from your raw sequencing read data. So eventually it will remove all the non-technical sequence and we are come up with only read sequences. So then the next step which is getting highlighted here, that is PCR clean. So what this PCR clean will do, that it will going to remove your PCR duplicates. As during during the generation of RNA-seq analysis, so we used to done the PCR amplification. So it will remove all the PCR duplicates. So then the next options that are getting alignment, once you got the clean reads, the next is how you are going to align your reads against a reference genome or reference transcriptome. As here our goal is to generate a gene expression data table from the RNA-seq. So we are mapping our reads against a transcriptome. So for that we are using this mapping algorithm, that is Bottai 2T. So here what it does, it will align your reads against the reference transcriptome. By clicking on OK, we will execute that step. Then after the mapping of reads on a reference transcriptome, next is quantification of your reads. So here we are using this RACM expression algorithm. So with this algorithm, we will able to generate a gene expression table on which you can perform various kind of analysis. So this particular RACM, which is also known as RNA-seq using expectation maximization algorithm. So this software will eventually quantify your expression in the form of FPKM, RPKM or TPM depending upon your input rates. So then here I am also executing the another analysis, that is differential gene expression analysis. So once we will have a gene expression table, then we can also perform differential gene expression analysis. So here we have different algorithm. Right now I am using the DSEC2. So it will perform the differential expression analysis between these two conditions, that is ER positive and triple negative breast cancer type. So then we can end this pipeline. By clicking on it, we will able to complete this pipeline. So once your pipeline will get completed, then under the download pipeline output files, you will get your all the outputs. So the first output that I am going to show you, that is how the gene expression table. So let me just download this gene expression table. So this is the gene expression table that we have obtained after running this DSEC pipeline. So let me just paste it in the Excel. So now we have this gene expression table where our samples are in the columns, while in the row we have ensemble IDs for the transcripts, again for the genes. So here you will get like ER positive sample, then you will have at the end you will have a triple negative sample. So this is the first outcome that we have obtained after running this pipeline, where we have obtained gene expression table in the form of FPKM values, where you can perform various kind of analysis. So then the second output that we have obtained, if you have observed, as we had also executed DSEC analysis, so we will also get the output for that. So if you download this folder, that is DSEC all pipe chips, so then we will able to obtain this folder. So here you will have a different output. So the first output I would like to show you, that is this table, DSEC table, the results from the DSEC analysis. As Dr. Mohit has already shown you, that the most important thing, point is identifying the important genes that can differentiate between two different conditions. So this is what the table we will obtain. So like here, we have first in the first row we have genes, then we have in the third row, like here we have log2 fold chains, then in the last column we have p adjusted value. So then we can identify only important significant genes by filtering out these genes based on the p adjusted value. So the p value which is considered as significant, that is p value less than 0.05. So you can filter out only important genes. So this is the another analysis that you can run. So apart from differential gene expression analysis and the obtaining the table. So once we have this gene expression table, so the other analysis that we can perform, that is we should able to normalize this data. So this is what the kind of gene expression table that we have obtained after running this RNA-seq pipeline. So during this program, you will learn various how you can execute various exploratory data analysis. But to perform any exploratory data analysis, your expression data must be normalized. So as it is well known fact that most of the statistics methods can be applied on a data that follow normal distribution. So when looking at like original data, here of this, a large number of samples will be look like outliers. So this type of distribution will interfere with many of the statistical tests or machine learning algorithm analysis. So to reduce this wide range of variation, we need to perform normalization of the data. So like after applying the normalization, we can actually derive a significant meaning from this data. So thus we will learn about different methods to normalize transcriptomics data, like log scale transformation or the quantile normalization. So next, to understand the pattern of the distribution of the sample within our data, we can perform various types of exploratory data analysis. So there are a number of the exploratory data analysis available, like number of the unsupervised as well as supervised algorithm. So let me demonstrate how you can perform principal component analysis as well as quantile normalization on your data once you have a table using this simple T-bar-info platform. So let me go again on T-bar-info server. So here under the area analysis, we can again direct it to the first interface. So here under the data mining, you will find different options like supervised, unsupervised, utility, factor analysis, then differential gene expression analysis. So as I'm interested to perform quantile normalization as well as PCA, so I'm taking the example of this demo utility PCA. So here already we have uploaded this data that we have obtained from the RNA-seq pipeline. So then by clicking on OK, we able to upload this data. Then the next step is quantile normalization. So then here we are mainly executing two steps. First, we are converting our FPKM value into the log scale, and then we are transforming these value into the quantile normalized using a threshold 5. So we are clicking on that. Okay, be able to perform quantile normalization. Once we have normalized data, now we can perform various exploratory data analysis. So right now I am performing principal component analysis, that is PCA. So by clicking on OK, be able to perform that. By ending, be able to complete this pipeline. So here under the output, we will get these results. Let me just show you this PCA plot results. Similarly, so here, so these this plot we can draw later as well. So here we have obtained the PCA components. Like here, the first component here, it contributes nearly 30% variation in the data. Then the PCA2 component that contribute nearly 14% variation in the data. So by taking this these two components, we can draw a PCA plot or scatter plot. Sorry. Just give me a second. Just give me a second. So then we can draw a scatter plot to understand the pattern. So like this one. So then we can actually mark them and plot something like this. So here, based on the PCA plot, we can visualize the difference between the two classes of the sample, that is ER positive and triple negative. Besides, because this particular data set also have mice models, that is a thymic and the knot, so we can draw the pattern for that as well. So this is how you can draw PCA plots simply using this PCA pipeline. And then the next platform apart from that, what are script coding, how this scripts are working behind. So then we have the another platform, that is codomics logic. So if you are on this platform, here under the courses you will find different courses how each of these scripts work. So the, for example, first I am going through the one of the PCA pipeline. So here in the course you will find the different lessons for each type of analysis, like data visualization, then dimensionality reduction, then differential gene expression analysis, and clustering as well. So if I go to the one of this lesson, that is dimensionality reduction by PCA. So in this, each of these lessons you will find a different subsections. So here the first step will provide give you the detailed introduction, like what you are going to perform. So then in the, once you able to complete that, in the second part, that is learn the code. Here you will learn how this script is working. So how you can write your own code. So what each step is doing, you will get that information. So once you're able to learn this code, in the next section, you will try it yourself. So for example, I'm taking this PC plot. Here first we are loading a libraries, that is a required package. Then we are loading our data. Each step is written with the hash, exactly what the next step is doing. So then we are taking only the data that contain without only numerical data. So then we are perform principal component analysis using PRC-com function. So then once we able to perform PCA, then how we can plot. For example, first we are going to perform simple PCA plot. Then we are also creating, we are also adding colors. Then we are also adding the frames, and then you can also add the labels, that is what are these samples. So by clicking on run, you able to execute this code. So like here we have obtained different plots. So let me just show you, like this is with this example data. So the first you will get like plot like this, where you can how your samples are distributed. So then you can also draw up frames against each of these subtypes. Like here we have different groups, like this is from the breast cancer cell line data, which have 4 breast cancer subtypes, that is basal, claudin low, luminal, and normal light. So how we can visualize the differences between these samples. So this PCA plot will help you to do that. So on codomics, you will learn to execute code. So then under another section where you can modify these codes according to your own analysis. So now let me go back to the presentation. So this is how you can generate your PCA plots. So this is about the codomics platform. So in the last week, to provide hands-on training and to make participants familiar with the approach to design and complete a research project, we are continuously designing different research projects using publicly available data based on studies published in high-impact journals. Thus, we have a collection of research projects where you can learn how bioinformatics can be applied in the field of various fields of healthcare using publicly available data to drive biological and clinical important signatures. So let me give you an example of the liver cancer project. Here we have explored different omics data types, that is RNA and miRNA in clinical and demographic data, employing multi-omics data integration approach for the liver cancer patient to find out the molecular signatures that are associated with clinical phenotypes of the liver cancer patients. As one of the major challenges with this malignancy is that a single omics data is not sufficient to design highly efficient biomarker. Thus, multi-omics data types can fill out that lacuna and will facilitate to design efficient signatures. But then another major challenge is the integration of those multi-omics data types. Either you need to develop an individual pipeline for each data type, then you will find an approach or a method to integrate them together. For this, you required high computational and bioinformatics skills. So to remove those obstacles, various multi-omics data integration tools like SNF, iCluster Plus, O2PLS on a T-Bioinfo server were integrated where users just need to provide input data set and can easily perform various types of multi-omics analysis by just clicking on options as I have shown you in demonstration. So in this project, we have also used these multi-omics data tools to find out important signatures. So finally, we have tried to multi-omics data integration that can provide us holistic picture of this lethal malignancy and how they are associated with diverse clinical phenotype like tumor stage, risk factors, age, gender of the patients. So participant can explore this project. Beside, we have another project on COVID-19. So in this project, you will explore about this pathogen coronavirus itself as well as the disease caused by it. Then you will learn about its origin and how much it is related with already existing human coronavirus strains using NGS data and bioinformatics methods. Next, you will also see what important components of the virus through which they enter into the human cell. And finally, you will see how you can identify important transcriptomic signatures that can significantly different under therapeutic treatment and untreated conditions. Then you will also get a project over Alzheimer disease. So as we know, Alzheimer is a complex neurodegenerative disease caused by several genetic and epigenetic factors. So these are driven by genetic variations, changes in the gene expression, as well as the other omics data types. So here you will learn on this project, you will learn how you can obtain a data set from public repository and execute different types of analysis. So in the later part of this training program, you will need to submit a research proposal. So here you need to define various fields like project title, that is your research questions. Then you need to define your exact research statement. Then you need to provide project summary, that you need to define what is your expected outcome, and then what are the different methods that you can employ, and then you need to provide the data set that you can use and as well as the reference publications. So in the past, there are a number of the research fellows who have completed their research projects in the diverse fields based on NGS transcriptomics bioinformatics with the Pine Biotech Research Fellowship Program. So among them, one of our research fellow, Mr. Kalaumari Maril, who is a PhD student from Mexico City, he's recently completed his research project entitled Classification of Triple Negative Breast Cancer Cell Line by Gene Expression Analysis of the Transmembrane Protein using various types of machine learning algorithms that are integrated on T-Bioinfo server and based on available cell line data employing these tools. Then we had another research fellow, Ms. Sime Dolanar, an undergraduate student from Turkey, recently completed her research project entitled Long Non-Coding RNA as Therapeutic Target in Chronic Lymphocyte Leukemia using publicly available data for CLL and employing different tools from the T-Bioinfo server. So I'm looking forward to become a research trainer, and now I would like to pass this session to Ms. Beepsa to talk about registration process and more about this program. So over to you, Beepsa.
Thank you so much, Dr. Harpreet. So let me introduce you to the different program resources as well as some of the free resources we have for all of our participants that you can start learning from right away. So for different parts of this 1-month program, we'll leverage Omics Logic resources that will include the asynchronous courses, as Dr. Mohit has shown, the cloud-based analytical server where you can analyze large RNA-seq data as well as visualize the results, as Dr. Harpreet was demonstrating to you. The Omics Logic code playground where you can practice coding in R and Python, as well as a repository of project examples. So to do this, the first platform that we have is the EduTBioinfo platform. And let us together go and see this platform. So please come with me to edu.t-bio.info. So in this platform, this is a free login. And once you log into this platform, you will have a membership to the free resources for a lifetime. So this is the EduTBioinfo platform. And here is a free resource that if you have not already taken, I would invite you all to go ahead. It's the Introduction to Bioinformatics course. And this course will introduce you to the different areas of application of bioinformatics. So if you are someone who is new to our bioinformatics community, we invite you all to go ahead and take this course. This course will give you an overview, and it starts with an introductory survey. This survey is very helpful for us to understand what are your areas of research interest and where do you want to go forward with the kind of bioinformatics training you're looking for. So please go through this course if you have not already taken it and help us learn more about you. And talking about what Dr. Harpreet has already shown you as part of this program, as part of the Omics Logic training, addition to the courses, you have access to different projects, right? So here is a project on tuberculosis that Manisha has been asking about. So you can see, so these projects are available to all participants or part of the Omics Logic programs. If you want to take this project, as you see, there is a cost for the projects to be taken if you're doing them separately. But for all participants of any of our programs, many of these projects are included depending on the kind of Omics Logic training you are going through. So there are different projects here on Ebola, on cancer that we will be taking examples for in this 1-month training. And the next step, as Dr. Harpreet shows, is to come up with your own project proposal. So as this is a 1-month training, we understand there is not enough time to come up with a full-fledged project. But what we would hope and we help you do is come up with your own project proposal. And I'm going to be sharing with you some examples. So these are examples from people and participants who have taken these kinds of projects and done it themselves. So these were all done in our research fellowship program, which is the next step, where once you complete a training program, the next step is to develop your own research project. So all of these projects that you would find on this page has been proposed by the participants, and then they have worked very closely with our mentor team to work on these projects. So you will see different examples from participants who come from different levels of education. So we have high school students who are doing these projects, as well as we have PhDs and postdoctorates who are doing these kinds of projects, utilizing public domain data sets to answer different kinds of biological questions. So you're welcome to go through this free resource. And going back, so what do you gain at the end of this 1-month training program? You will receive a certificate. So we give out 2 different certificates. Here is an example of a certificate of excellence, which was given to one of our participants from our previous program that we had in last year. So the certificate of excellence is given to anybody who has over 10,000 points. So all of the courses and the server platform as well as the Codomics Logic platform are connected to each other. So we are able to see your progress over time and learn more from you and your activity, like what you're doing. And by that way, we are able to determine your progress and work with you to help you learn more from this entire training. So we also give out a certificate of completion, and the minimum requirement for that is to at least have 3,000 points, which is very easy. You would have to do a few courses and run a few basic pipelines to get those points. And then what I have here are examples from participants who have already gone through trainings like this. So Urja here, you will find her example under the project example link that I have shared. She has completed her research project on differential identification of biomarkers between neural stem cells and glioblastoma stem cells, and done that project, and is now going forward and presenting that project at different student conferences, and also helping and working towards her own publications. So these are the different opportunities that you have. So what you do with your project is completely open and up to you. So you are able to take forward this training, whatever you are learning through this experience, and then explore the different areas. You can go and apply for a new position. You can start learning more about RNA-seq. You can apply the knowledge to a project. So you have several different examples of other participants who have taken these training and then gone ahead and applied them, won competitions. For example, Anurag collaborated with Arva Bora. They both were part of a training program. And then they went ahead to win a pool team challenge. So there are these different opportunities which are available to you. So you all can go ahead and explore. And now I would want to invite you all to go ahead and also start joining us on our social media. So if you're already there with us, linked to our LinkedIn page and our Facebook group. So let me share the links to both of these pages very quickly in a minute. Because in our LinkedIn page, if you see in our LinkedIn group, then you'll find that there are a lot of participants who are joining us from all over the globe. So if I see everybody here, there are not only students who are joining us, but there are also other researchers, there are also faculty. So it is a community of bioinformatics interests. It's a community of people who have similar interests. So you are able to meet together at one place. So this is the LinkedIn group, and I'll post it again in the chat box for everyone who is interested in joining. And we also have a similar group in Facebook for those of you who are interested, and I will be posting this again in the chat box. So please go ahead and join these different groups. And now regarding the question about how to join this training program. So this is the link of how to register for this training program, and I'll also explain the different steps. So I'll post the link again in the chat box here. So if you go to this link, you will be able to find the different syllabus for this program. It has all the program dates and the different topics that we have also covered in this session. And if you click on Get Started, you will be given 3 options. And let me explain to you what the 3 options are. If you are just a beginner and just getting started with bioinformatics and learning about NGS data analysis, we recommend you the beginner option. This covers all the basic training program, as well as the research projects and giving your proposal during this entire 1-month training. While the intermediate option comes with one additional aspect, which is you get mentor support during the part of the training, whereas you can request one-on-one mentor sessions beyond a group session. So we have these different dates, as you saw, where we will be meeting in an online class like this and talking to you about different topics. So beyond those sessions, if you are interested in taking extra mentor help, you can also go for that intermediate license. The advanced license is recommended for anybody who wishes to work on a project. The advanced license gives you 2 months instead of just 1 month. It gives you 2 months, which is an extra 30 days access. And you will also get an advanced license where you are able to work with your own data sets if you want, or you're also able to work with our team of research fellows as well as our own omics logic trainers to work on your own projects. So this will give you the 2 month option. So these are the different payment option. We will also share with you some discount codes which are available for the program, and you can register for this from right here. And we also have another option for students and participants who would require some kind of scholarship. So we do have a scholarship option as well. So if you are someone who requires the scholarship, you can also fill up the scholarship form at the bottom of the page. We do have some prerequisites which you need to offer. We will meet with you, we'll discuss, and we only offer these scholarships to students who are deserving. So we'll have that meeting with you and we'll decide whether you get a scholarship for this training program or a discount. So we'll let you know as we do the scholarship meetings with you. So these are the different ways you can opt for this training program. And once you are in the training, you will gain access to a page like this. It's called a forum page, which will have all the additional uh, details about how to get to the different aspects. So this is just an example of what the forum looks like. It has the event dates. It has some description about the program. You will also get to learn about the other participants who are joining along with you. We welcome collaborations and we hope that you are able to collaborate with each other on projects, on discussing about different topics and make it a collaborative environment. And we have a forum option here so that all the participants besides the sessions are able to come up and ask their questions to their mentor team so that they're able to answer those questions for you, post any other interesting topics that you find. We also share the top participants here from time to time. So these are the different aspects of the training program. And we are also able to see the different progresses for different participants who are part of a training from right from this organization page. So I welcome you all again to go ahead and join this training program. I'll be posting this in the chat box. And if you have any questions, please reach out to me at marketing@pine.bio. And now I would want to ask you all, if you have any questions, please let us know so that we are able to answer those questions for you. So is there any specific question that you would want to ask us or our team here, and we can answer them for you? So the chat box is open for questions, and you will also be able to unmute yourself and ask any questions that you have to our mentors directly regarding any of the aspects that we were talking about today. So please post your questions either in the chat box or let us know. We hope that this program is going to be helpful for both students as well as clinicians and researchers. And this is the reason we have the training designed this way, where we start with very basics, so that if you just have a very basic idea of cell and molecular biology, you're able to join the training, understand those basic concepts, and then build up slowly, given with the different tools and resources that we have shown you as part of the training program. And I hope I don't see any questions today. So I want to thank you all for joining this session. And yes, the timing for the program is 5 p.m. Indian Standard Time, which is the same time as this webinar today. So you are able to see all of the different sessions starting from March 2nd. The different sessions, all those sessions will be on 5 p.m. Indian Standard Time to 6 p.m. So if you have any other questions related to the program, you can ask us again. And thank you all for joining today. So Manisha, you're asking, can the timing be 5:30 p.m. IST? For now, the sessions have already been designed. So if you cannot join the session on time, is there any other option? Of course. So what we do is that, like today's session, we record all of these sessions every day, and we have a private YouTube channel where we put it. So only participants have access to those links. We share it with all participants after each of the session is over every day, along with the assignments and any other details that we have discussed during the session. So in case you're interested in joining this training program now, we will keep it open and you can actually go forward and join the training session. And if you're missing out on some of the classes, you will receive the recording for that, as it is a 1-month training and we'll go week by week. So you will be able to learn from these different sessions, and you can also review them at your leisure during the weekends or any other time that you have. And we'll definitely consider if you have any other requests regarding the timing. So you can, if a lot of people agree that it could be done at a different time, then we definitely can. So we had actually done a poll on Instagram where we got this idea that many people are free at around 5 p.m. Indian Standard Time. So we had done that last month and we got that answer that a lot of the participants are interested in doing a session at 5 p.m. So we asked our community and we got that answer. So we have designed the sessions to be at this point of time, but we are definitely open to any other requests that you may have and we'll try to consider them. So I hope that we have been able to answer all of these questions. We'll have more webinars as well as more sessions in the upcoming weeks. So please follow the email, which is marketing@pine.bio, for any other information regarding this. And I thank you all again for joining today's session. Have a lovely weekend, everyone.